speak a little bit to the pre-IND meeting with FDA in terms of how much of a dialogue versus a dual monologue it is, and help you design your large animal studies to avoid any oopsies down the road when you get to the point where you want to file the pre-IND where they don't go back and tell you this product or that protocol was not approvable.
Mm-hmm, mm-hmm. So, the challenge is always how early do you go and how much information do you have to actually engage in that dialogue. So you need enough information so that it'll be a useful, they can't just say it depends, right? So you need, information and sufficient enough. So generally, what you need is actually a clinical trial outline, something, they need to know where you're going. As Ellen said, you need to know where you're going. So where you're going early on is the first in human study. So you need to understand at least, your clinical trial outline, what the population you're going into is, and you don't have to say, well, we're going to do these different dose levels, but you can say we might, we're going to use 3 cohorts and whatever. So they need to understand that. And then a little bit about the cell. So, or are you talking cells or the product? Okay. So basically, not the final manufacturer, not a GMP manufacturer. It could be research grade or whatever but some characteristics of the cell. Okay. Obviously it's derivation where it came from, but a little bit about the characteristics. How are you characterizing whether or not the phenotype of the cell or any information that you have in terms of you'll have early studies now on proof of concept or in vitro activity to say, okay, this is how we think we're moving. One important thing to understand is whether or not, the activity is based upon just a paracrine, autocrine type of versus, actual engraftment, and you have to, the cell has to persist, whatever. So if you know anything about the mechanism, and then you would outline, well, these are the studies that we think we're going into. We're going into rats. We'd like to go into rats. And the agency is not keen on developing protocols, so you'll have to put together some straw man proposal as to what dose levels you think you might need. And you don't even need to know exact dose levels. You can say low, mid, or high, placebo, whether or not you're going to have a positive control, et cetera. Something about the design of the study so that they can actually provide commentary on. You'll know whether or not you'll need an animal model of disease, or you'll say, these are the limitations in my animal model of disease. This is what I'm going to do. I think it's a more difficult discussion to go forward to say, okay, I'm developing this product for X disease, but there are no animal models.
I just want to make one comment to the question. We actually published a paper sort of on the who, what, when, and why of when to interact with the FDA, and it was published in October in Stem Cells Translational Medicine. So when I put the slides together, I'll put that reference to the paper because you might find it useful. It talks about the pre-IND, which actually is not something you'll find in a guidance document. It's not part of the formal meetings. It's actually always an informal discussion, and it's not at every center. It's generally just at the Office of Cellular Tissue and Gene Therapy. So it's actually hard to find. So at any rate, as you noted, it's a big focus really on the preclinical and the manufacturing, but they do want to know where you're headed in terms of the clinical plan. Thanks.
That's a quick follow-up. How receptive has the FDA been to iPS-derived cells, autologous iPS-derived cells?
I say you can never say never and never say always. The agency sees everything, and they're not going to come out with you cannot do, right? It will take you longer, right? With anything novel, again, novel is scary. So there aren't any guidelines. When I first went to the agency, I was in an office, and then people started calling me about everything. I'm like, there was nothing to pull down to answer the question, right? So, or when people decided, to make proteins with transgenic animals, what are we supposed to do? We had no clue. So actually, we had a Keystone conference, and manufacturers, it was a fantastic Keystone conference where we had manufacturers making proteins out of transgenic goats and pigs and whatever to talk down and say, okay, well, this is, good animal practices, right? You have good, we had no clue how to regulate these things. So I will tell you that the agency, there isn't a book on anybody's desk to say, okay, wait a minute, iPSC, okay, yeah, this is exactly what we have to do. That's the challenge, right? you know more about your product than they do. I mean, within the Center for Biologics, I mean, and I don't know how long it's going to last, but there are people who actually do research. So, I mean, that's great. That helps us with some of these novel therapies, right? But you know more, and the challenge is to tell the story. You need to tell the story that they understand, and the limitations, et cetera. And then it's almost a kind of a co-learning type issue. But again, as Keith said, there are no absolute. Look, people wanted to use tumor cells to make vaccines, right? I mean, they had workshops. I mean, when it gets to be at that level, at a level where now a number of people are doing it, that's when you'll see the guy, you know, I mean, there's a draft guidance that's out for comment for preclinical for cell-based therapy, gene-based therapies. We've been doing gene therapy, right, since 1990. That was the first therapy. There was an earlier guidance on gene therapy, and we've been doing cell-based therapy. So now this is kind of an evolution of that. But it's just starting to mention cell-based, but that guidance is not very specific because there isn't any specificity. So
our group has gotten a pre-IND for iPS cell-derived keratinocytes, and the FDA group has been very encouraging, very interactive, very helpful with suggestions, and they really want the therapies to work. They want it to be safe, and so if you come in with a plan and know a lot about your product and your target product profile, they're very encouraging. They set the bar high, but they're very encouraging. So I think that's a encouraging place to be.
Right.
Yeah, just one other thing. If the FDA doesn't have a guidance document specific for something like this, they'll try and find the next best thing. All right, so that's something you ought to take into consideration. You know, find some other guidance document that, you know, while not dealing specifically with that issue, might give you some guidance in how to approach it. So that's the other thing that you might want to try.
Right. But I will say that for a novel therapy or something that, if there is an article out there written by someone that is actually scary and says something that you don't think is right, you better do some splaining, okay? You can't afford, you know, I mean, because that's going to be a challenge with anything novel. I mean, if there's a group out there that's developing something that you question as a scientist, better that you try to put it in some context than have the agency start worrying about something that may not be a problem, that you don't think is a problem. It's not related to your product, so I don't want that baggage or whatever. So I think that's one message I would say with respect to novel therapies is you need to scour the literature and make sure, because the agency is pretty good at finding those kind of one-off papers that probably you don't want them to find. So, you know.
So my question is on doing GLP studies for the pivotal safety studies for preclinical, and how much, as an academic trying to do on a budget, trying to do an important study that provides the dose range for clinical studies, how much outsourcing do you do to companies? How much just to get enough data for the pivotal study?
So you're talking about doing a pilot study to support the pivotal, or
doing the pivotal study? How much? I mean, in terms of recommendations for path for the necropsies, and you're mentioning that you opt for some of it, and so in terms of recommendations, more optimal types of combinations that give you the necessary pivotal data.
So is it a novel procedure that to introduce the cells? Do you think things that? Then, you know, I think probably the best place to do it is where you're, you know, I mean, some people take their surgeons to the site. So, I mean, so you can go to a CRO and take your surgeons to the site. The surgeons could do the, you know, the treatment, or, I mean, the application of the cells, et cetera. So then you would do it all there. You can, you know, as I said, there are cases where the cells are administered and the animals stay at the site, the academic site. And then prosectors come in. The reason why you need more people is doing a necropsy is very it's just really laborious. And you're going to have to do biodistribution and that is something that is, with all those organs, and it's very, so I would never advise anybody to do necropsies by themselves, but certainly cell application is perfectly fine. And, again, the most important thing when you do the study is to have a protocol, and you should have a study director, and you should, all those things about the GLPs that state what it is and what it's not. Yeah, so I think, it's certainly fine. And this group is very accommodating in terms of GLP. And I would submit to you, had nothing, unfortunately happened in terms of the gene therapy situation, we wouldn't be talking, we wouldn't have, you only talk about things when there's a horrible thing. T-Gennaro, we've developed many monoclonal antibodies without a T-Gennaro, and then we have T-Gennaro, and then now we're revising how we extrapolate to the MABEL versus the PAD. We've been doing monoclonals since the 80s, and we have not had a T-Gennaro, so,
So, can you give us some insight as with regard to the immunotolerance issues? The FDA must have gotten a whole bunch of proposals involving human mesenchymal cells, human bone marrow cells, ES cells, human iPS cells, put it into a pig model, put it into a dog model. And, the different investigators going to say that we're going to use cyclosporin or cyclosporin and prednisone. What do you guys look for in terms of the readouts of the biomarkers to show that the immunosuppressive tolerance protocol that the investigators are proposing are effective or not?
So first of all, we guys, does that mean?
Previously.
Yeah, I know. See, it never happens. Yeah. I left the agency in 1997. Yeah. I still cannot. I still have to give the regulatory lectures all the time. So with respect to immunosuppression, are you asking whether or not, so you'll propose a certain, I will tell you that whenever cells aren't there at the end of a study, the first thing the agency is going to say is you don't have the proper immunosuppression, et cetera. It's a challenge. When you have cells or not, now, that's why you need to understand mechanism, because it may not be that they have to be there, okay? This is the biggest challenge for FDA, because they think that cells are being rejected because they're xenotransplants, and that in the clinic, the cells are going to persist forever. So the level of immunosuppression, whether or not there are recommended immunosuppressions, they want to make sure that doesn't impact. You have to understand that the immunosuppression is not going to be a positive thing. It's not, there are some concerns about immunosuppressive agents actually being active. So you have to show that the activity is related to your product. And then in terms of, our biggest challenge right now is to do large animal immunosuppression. But this is something that I think industry can help too, or academics. The good thing about academia is that you are and hopefully you share better than industry. So, that's really a challenge. Can you share more in terms of successes with respect to immunosuppression? The agency really can't, unless, they don't make those recommendations. They'll tell you if there's a safety issue, then somehow you'll be informed of that safety issue because you'll ask you to do things that you don't know why they're asking you. But if something works, they're not going to tell you that this works better than this in terms of immunosuppression. But, I think this is an opportunity for the field to be better understand if extrapolation is important and if a large animal can be, and how can we leverage some of the information? We're trying to do that with gene therapy and biodistribution. We're trying to say now that biodistribution studies, we know where ad vectors go, right? They go everywhere. No matter where you put them, they go everywhere. Not a problem. Can we stop doing biodistribution on the ad vectors that we have in experience base? And we're actually publishing in Jim Wilson's journal, just to kind of get it going, that maybe we don't have to keep doing. So once we learn something, so, MSC cells, once you know where they go, and if you give them IV, does everybody have to do biodistribution with MSCs? This is, the field has to move towards making recommendations, as a group, and, putting position papers together. So, and maybe, one position paper is how best to immunosuppress.
So I had a question about designing preclinical studies right here in the front. And it has to do with behavioral studies, because as academics with animal models of disease, we often focus on behavioral improvement or outcomes. And I went to a drug discovery conference last month, and I was really struck by how much behavioral outcomes were being de-emphasized. And that was because when they talked about how non-predictive the animal studies were for human, the thing that was the most non-predictive, according to these studies, was behavior. And the things that were more predictive were what they referred to as the 3 pillars, the target, engagement, or exposure, engagement, and activation. And they were focusing so much on that, and they were de-emphasizing behavioral outcomes in their preclinical studies. I was wondering if that was a general thinking in the field that there might be a shift away from focusing on behavior.
Yeah, I mean, I don't know how you extrapolate, a signal pathway to a dose. Again, I think behavior helps you with the dose and active. So what can you get out of this? It may not predict exactly with the clinic, but at least it tells you that you have an active dose that's doing something. I don't know how to extrapolate from a signal pathway. I mean, so all I think about is dose. How do you that's where we have to do it. And can you get a dose out of in vitro? Can you get it out of understanding the pathway, engagement, a target? I don't know how to do, from a concentration to that. So maybe it's I would never, again, I say never say never, never say always. Maybe it's a composite. Maybe if it's imperfect, if behavior is imperfect, that gives you one piece of it. And then now you do the target, and, it's to help you with the dose. I don't know how you get to the dose that way. Maybe they have an idea. I don't know.
Hello. Like you said, novel is scary. So I was wondering, there's like the clinical trials website, I can find out where the clinical trials are. But when these data are submitted to the FDA, is there any place where there's like non-confidential data can be reviewed by other people so that we can...
So again, not with industry, but the NGVL, there are some gene therapy folks here. The NGVL, and now it's morphed into something else, but I don't know because I was involved with NGVL, created a database for NIH-sponsored studies where you would actually put your preclinical data. Mm-hmm. And again, Jim Wilson's... can't remember the name of the journal. And I think the stem cell translation is open to publish these studies. I talked to Mahendra. He said that. So you just have to publish it. The agency cannot publish these data, cannot give you access. Okay. It has to be you all, doing the studies and publishing, and in as much detail as you can. Offer. It's a challenge, right? It's a challenge. The agency cannot talk about any of this, and so how best do we move forward? And it won't come from academia. So I want to tell you just one example of a field that went nowhere because what happened was it went into corporate America, and that's the artificial blood, the substitutes. Toxic because there's something teleological about hemoglobin being in a red cell, and not just free flowing. But anyway, that was in the Army and the Navy and academic institutes. And then when they came into companies, then all of that became closed, and all that toxicity was nobody shared. And so basically, we have this was 30 years of research, but. So if we sequester, it's a good word these days, or a bad word, this information, there's something about sharing that it advances science. And in this area, it's really important, so.
And I have a follow-up then. You said that the FDA cannot publish, but they can certainly guide us. I guess, for our very specific example is we are trying to put cells into articular cartilage defects. Uh-huh. And in our case, you talked about a dose response, and in our case, it would be, different amounts of cells to see whether we could get the same effect using different amounts of cells. Now, we know that, clinically, there are therapies already like ACI that put cells into these defects. Mm-hmm. And so they have a little range of efficacy. And, we would like to relate it to the FDA and say, we should be putting in this much. But then,
you can, so that's Carticel. You can get some, Carticel was one of the first cell therapies approved, right? Mm-hmm. Not quite approved. Technically, it went through a different type of a process. But, So, I mean, that's exactly where you would get some of the information now to start leveraging that. I mean, the Edmonton protocol, when we talk about diabetes, the information that we have in earlier studies we use to help inform the dose. But that's precisely where you would go to try to use the argument to justify your dose. But that information, some of the information that you can get under FOI, once a drug is approved, but certainly not under, not when it's, not when it's in development.
Got it.
So, and again, the website, I don't know if, and it could be on the website. If you go to, well, at least for drugs at FDA.gov, you can find information on approved products. And biologics, if you go, I don't know if Carticel is there. I don't know. Do you know, Peter? But if you go to the Center for Biologics, CBER, just Google that and look for approved products. See if you can find Carticel. And it is there.
Yeah, you can also try looking up things like summary basis for regulatory action. You can try that on that the FDA website. And it's heavily redacted, but you might be able to glean some information.
It really takes out the CMC. You can't get anything on a CMC, but you generally could get information on the preclinical.
Vast tracts of nothing.
Yeah, I know.
And
I don't even know why
a miracle
they put it.
happens, and then.
So, is it true of this urban legend that it's easier to get investigator IND from a university than industry IND from the FDA standpoint?
Right. So the way I see it is that, I think the difference is that, one, as I said, the minimalist approach. There are some companies who have a bar that you cannot go forward into the clinic. So some companies, because the liability is different, so I think it's related to your risk tolerance, right? So for some companies, they will not go into first-in-human trial without this much. Certainly, the smaller biotech companies don't go in with that much. And again, that's not a minimalist. It's because they can't afford it, and it's very targeted. So I call it a targeted approach, right? I think the difference in terms of investigators is the definition of investigator and the preciseness. It's a single investigator, a single site. It's a very, versus, something that's more broader based. It might go to multiple sites. And so I think the perception that it's less is because, one, investigators don't do anything more than they have to do. And two, I think the agency does look at it as an experiment. Generally, the number of subjects are fewer. It's at a single site. So insofar as any of those concerns minimize potential risks in terms of even, transfer of material to, the different aspects that you have when you're running a clinical trial in multiple sites versus a single site, I think it takes into account. So, I think if corporate, I think that might come from industry to say, well, you let investigators get away with so much less. Some of it is because they didn't have to do as much as they did.
So let's say, for example, if investigator trial, the FDA approves 5 patients, everything went smoothly, and another company comes by and say, hey, I'm interested in doing the same study, but I want to make it a bigger scale. Does that company then have to repeat a lot of the studies that the investigator did before, or they could kind of take upon the previous study submitted by the investigator and just scale it up?
Oh, well, that's a little sleazy way to go forward, actually, right? So the company uses an investigator to get something easy, and then, I never liked that approach, but anyway. Certainly you can leverage the data. I mean, that data is, if the company owns that data, or if the company has full right of reference for that information, you would have to, if you have an IND, then they would have to have the right to reference the IND. So if that's information, then they can leverage that information. But they can't leverage, just because you submitted something, they can't say, well, so-and-so did it, so I'm going to design my trials. It has to be very clear that that company has access to all your data, all the investigative data. And then, of course, you can leverage the data. Yeah.
My question is that for autologous ex vivo expanded adult stem cells, in terms of heterogenic study, what the bar is? Because for this, there's no animals that can be used. Well, we can use new mice, but how much do you need to do in order to do that? Because this is not iPS-derived cells, not ES-derived cells, not autologous cells.
Yeah. Well, I mean, I think you have to show, again, the CMC, that you're making lines, or however you define what you're calling these cells, right? So these autologous cells, refer to them, I think some people refer to them as lines. So you make a number of them so you can show consistency of the process. So you need to make a number of them so that you can show process consistency. And then if you have an animal model, then you test a number of them in the animal model. And I think, again, when I was talking about setting specifications early on, what you want to make sure is the best thing that can happen is that you have differences in your specifications within the cell line. Because what you don't want to happen is very early on in development, you assign your specifications for your autologous cells here. And now you have to scale up, or you have to do differences in processes you might change, and now things change, right? So ideally, you'd like to do a number of, whether or not it's 3 or 5 or 10 cell lines that gives you the widest range to say, this is my product. Because if you only do 3 and they're very close, then you have to say, this is my product. And the next time you make it, right, if it comes out to here, now you have to justify why that's the same as this. So I think you should make enough cell line, I think 3 is too small, right? And I don't know how many you recommend to characterize the process, actually the wider the specs gives you more flexibility. The wider the differences, in some degree, gives you more flexibility over your course of development, because then again, you're not narrowed by saying I've qualified this spec, that you have animal models that actually have tested cells, wider.
Yeah. Generally, as you go further and further in development, the specifications tend to narrow. They don't go in the other direction.
Yeah. So.
Yeah. One more question is that in Europe, the treatment has been done without the previous regulatory, by the European groups. Can we use this study, use as a standard, or how much data can we get from the European, publication? There's nothing US. So how does it go about that?
So the study was a clinical study or a preclinical study, or what?
Retrospective, collection of this treatment have shown such an effect, but that was not done under the standard regulatory.
Well, I mean, again, I think, so you're saying there's clinical data with a certain product where you have some information, but it wasn't done according to GCP.
Yes, that's correct.
So, I mean, I think obviously the agency will look at it and it'll be supportive data, but it won't be, and I think actually it was probably closer to a dose than to go from the animals, right? So you'll at least have some information about dose. Whether or not you can start with the highest dose that you use, or whether or not you have to go back a cohort and try to understand the dose. I mean, again, they won't have the real data, and then it depends on how much data you can bring forward. If you can bring any original case report forms from those studies, then you can use more of that. If it's only a publication, you're probably not going to be able to use too much, except you'll have a better justification of a dose.
Okay, well, thank you so much. We're going to have to move on to the next group of speakers.
Okay.
Thank you very much.